Okay, good afternoon, everyone. My name is Cynthia, and I am a third-year PhD student in the Advanced Drug and Gene Delivery Lab in the University of Iowa. And this afternoon, briefly, I'll be talking about an evaluation of bacterial cellulose that we did to investigate if it has any intrinsic adjuvant activity. And this is the outline that I would follow. First off, I just want to start my introduction by defining or mentioning what cellulose is. It's the most abundant naturally occurring polymer on planet Earth, and it can be sourced from different organisms, including plants, bacteria, fungi, algae, and one animal source, which is the tunicates. But for the purpose of this presentation, we'll be focusing on bacterial cellulose. And the reason we are doing this is because we know that bacterial cellulose has a lot of amazing properties, including its high purity compared to, for example, plant cellulose, which when obtained from plants would usually come in a mix with hemicellulose, pectin, lignin that would have to be taken out before the cellulose could be utilized for the purpose for which it's intended. And that's not the case for bacterial cellulose. Also, some papers talk about the fact that bacterial cellulose has an architecture that mimics what we have with the ECM. We also see that bacterial cellulose is able to hold up to 200 times its dry weight in water. And when it comes to mechanical strength, bacterial cellulose also has high mechanical strength. And because of these properties, we find that bacterial cellulose is useful for some potential applications in biomedical science, such as drug delivery, biosensors, wound dressings, and others. In terms of FDA approved products, we only have about 2 products that have been FDA approved that are sourced or that are made of primarily of bacterial cellulose. The rest are concepts in the making, ideas, and papers that have been published saying that bacterial cellulose has potentials in this area. And so all things were going well until we came across this paper that mentions that bacterial cellulose has the potential to serve as a vaccine adjuvant. Now that becomes a concern because we don't want to use a biomaterial that would eventually be a source of immunogenic reaction. And this matters because we have seen cases, for example, with this textured breast implant that was recalled in 2019 following a tracing or some link that they saw that this implant was leading, the material being used for this implant was leading to a rare kind of T cell tumor, a lymphoma was resulting from the use of this particular breast implant. So we don't want that kind of scenario in the case where we start using bacterial cellulose as a biomaterial, for example, for tissue engineering or something in that light. And so this matters because if indeed bacterial cellulose does have this adjuvant activity that this study has proposed, then it would mean that it would not be suitable for some or all of these biomedical applications of bacterial cellulose that are being tested. It would be a waste of money, a waste of time, and a waste of resources of scientists, PhD students, PIs who are currently working to see that this is made possible. So for our hypothesis, we wanted to see if truly bacterial cellulose would increase humoral immune response, or will it increase cellular immune response, and will this match or surpass the performance of what we already know to be the gold standards in terms of adjuvants? To begin with, I would like to define a vaccine because I know we have like a broad audience here. So a vaccine is a biological preparation that stimulates the immune system to produce a protective immune response against specific disease. And adjuvants are substances which, when added to the vaccine, would improve the immune response. So in a case where a vaccine is administered, we do see that there would be antibodies being produced, there would be production of memory B cells. However, if in the case that adjuvants are being introduced, then you see cytotoxic T cells, you would have more antibodies going into the system, and you would have more production of memory B cells. So the first aim of this experiment was to produce and characterize bacterial cellulose. And to get this done, we used the K. xylinus culture in HS media, had this on for about 4 weeks, and then got the bacterial cellulose, which we were able to dry by open air. And then the next step we took after that was to carry on with the characterization of bacterial cellulose. And here what we wanted to do was to see that it matches the standard in quotes for bacterial cellulose based on what other groups have already published. So what we are looking out for here is the XRD patterns to see if they have similar peaks with what has been established already in literature, the type of fibers that are being produced, the diameter, and how it matches what has been established in the literature, to see if it has the functional groups expected for for cellulose, and then to also study its thermal stability. So after having this done, we went on to the next step, which was to sterilize bacterial cellulose, remove the endotoxins, and then reduce particle size before we can carry on with the injection. To get this done, we did endotoxin removal by a combination of heat and alkali method. And after that, we went on to quantify the endotoxin levels, autoclave, and then homogenize bacterial cellulose since it came in a sheet and we need it to be in at least at most microparticles for it to be injected for this experiment. At the end of the day, we had 0.52 endotoxin units per milligram of bacterial cellulose. And each time I present this, everyone always wants to know what the FDA limit is for endotoxins in parenteral injections, and that would be 5 endotoxin units per kg of body weight per hour. However, this is for humans, and this study was done in mice, and so you can't directly juxtapose it because mice can tolerate 100 times more endotoxins than humans. This endotoxin removal is a very important phase for us because we are working with a gram-negative bacteria here, and so by all means would have this outer membrane that is composed of the lipopolysaccharide region. And this lipopolysaccharide region has the lipid A, and that is the center of the toxicity. That is the main problem in question here. So we wanted to take that out because by all means it would give a false positive moving along with it in this reaction. And to be able to take it out, to be able to quantify it, we use the limulose amoebicide lysate. This LAL test is derived from the Atlantic shoehorse crab, and the way it works is that these crabs on their own, their biology includes a clotting mechanism that prevents the spread of bacteria or fungi in the crab. And basically what happens is if they are being attacked by gram-negative bacteria, for example, those lipopolysaccharides would orchestrate this cascade of events that would eventually lead to the clotting of whatever intruding organism that was. And so in the same vein, that's how this testing works. If you have endotoxins in the system, it would stimulate this cascade of reactions that would eventually lead to either a coagulation that you can now see increased turbidity in your test kits, from your test kits, or you can see yellow chromogen release, yellow coloration, which was the mechanism behind the kits that we used for this experiment. Next on, we went to do the particle size analysis after we had homogenized, and we saw that we had about 2 micron size of particles. However, there was a time-dependent aggregation over time, and this DLS data was derived after 10 minutes, while this was derived after 24 hours, the SCM data. Next, we went to actually do the vaccination this time. And to do this, we introduced ovalbumin, which was the antigen for this experiment, and then administered these to mice and did a tetramer staining to be able to identify the CD8 positive T cells, and then ELISA to see if any antibodies have been produced as a result of this treatment. We had 25 animals in total, 5 animals per group. The first group did not receive any vaccines or any treatments at all. The second group received just the antigen, which is soluble over. The third group is our main test group that received bacterial cellulose and over. And then the fourth group received aluminium hydroxide and over. The aluminium hydroxide is the oldest known adjuvant and is still gold standard till today in terms of being able to produce IgG1 antibodies. And so we needed that as a positive control for our ELISA. And then the Ad5 over is an adenovirus that would deliver over to the cells, and it's something that we have used over time in our group to see that this produces over-specific CD8 positive T cells. So it will be the positive control for the tetramer staining experiment. And this is the vaccination timeline. This is the vaccination and sampling timeline. We took blood out on day 14 to be able to measure the over-specific CD8 positive T cells, and then took blood every week from day 21 to day 42 to be able to measure the over-specific antibodies. To be able to measure the over-specific CD8 positive T cells, we anesthetize the animal and then drop the blood into ACK lysis buffer. And then from there, we went on to do our staining, followed by introduction of the cocktail of antibodies that would then help us to identify the CD8 positive T cells, over-specific CD8 positive T cells using flow cytometry. While for the over-specific IgG, what we did was collected same blood, but in this case, we allowed it to sit for about 1 hour and then separated the clots and then did a centrifugation to be able to get the serum, which we then diluted and used for the ELISA. What we did find is that bacterial cellulose did not significantly increase over specific CD8 positive T cell responses. And we know this because we had the AdOver, which gave a positive response in this experiment. And then for the IgG1 and IgG2 titer values, we did see that as compared to alum, bacterial cellulose, which is our main test group, had low or almost insignificant titer values as compared to what we had for the standard. And this was the same across board from day 21 to day 42. We also see that for AdOver, which is the positive control in this case, it was also significantly higher than what we had for bacterial cellulose. We also did see that the treatment groups did not affect body weight all throughout the period of the vaccination of this throughout the vaccination period for this experiment. And so from this, we are able to conclude that bacterial cellulose and ova did not significantly enhance cellular or humoral immune responses, and that under the tested conditions, unmodified bacterial cellulose would lack those intrinsic adjuvant activity. Going back to the experiment and the paper that had published saying that bacterial cellulose has these adjuvant properties, we did see that they only did in vitro experiments and they also did not take out the endotoxins at all. Now, this paper wasn't published in the US, and if you were to do such an experiment in the US, anything with endotoxins would not fly because endotoxins were discovered as adjuvants right after we had alum being discovered, and till date, the FDA has not approved endotoxins or anything containing LPS to be an adjuvant. So we're glad that we're at least able to take that out and show that unmodified bacterial cellulose does not have an intrinsic adjuvant activity and so should be considered safe in terms of being used for biomedical applications that we have in mind to the best of our ability, to the best of our knowledge under these tested conditions. And I would like to acknowledge my PI, Dr. Salam, and all the Salam lab members and all my committee members, Dr. Shaw, Dr. Jadnik, Dr. Gary, and Dr. Shunhui, and her student Binayak for helping with the production of the bacterial cellulose, and NSF EPSCOR for the funding for this project. Thank you very much.
This is very interesting. Thank you. Questions for Cynthia? So when are you going to submit this for publication?
So I already have the manuscripts drafted and we are undergoing edits. This is like the 5th edit, but yeah, undergoing edits to get it published.
Where do you think you'll submit?
Cellulose Journal. Yeah.
Nice.
And thank you for Cynthia to include Binayak and myself in the manuscript. Thank you. I will send you my feedback soon.
Okay, thank you.
Can we talk about the horseshoe crab blood and why is it blue?
I used to think that was the only animal, but it turns out that the octopus also has blue blood. They're actually some aquatic animals that have blue blood. Something about them not having the regular red blood cells, I guess.
Yeah. They have blue blood cells?
They have blue blood, yeah.
Interesting. Other questions for Cynthia? What was the most surprising part of the project for you?
For me? Well, I mean, I feel like it's the fact that somebody thought that bacterial cellulose, I mean, it's good thinking because there are actually some glucans that are able to give that immune response, but those are beta-1,3 glucans. And then bacterial cellulose or cellulose in general is a beta-1,4 glucan. So it makes sense that somebody could think that, oh, if this can cause an immune response, would cellulose also cause an immune response? But if you don't take endotoxins out of the system, it's not something usable in the US as an adjuvant, no matter how good the study is. So yeah.
And those crabs, I'm sorry, like are they alive when they're draining their blood like that?
So I did read online that in one of the places where, first of all, there's now like some regulation that doesn't allow just anybody to go fishing to get those crabs to, withdraw their blood for that. But then in one of the places that they use, I think it's Florida or California, I think it's Florida. So they have regulations whereby after the blood is withdrawn, and it's specific companies that have the license to do that. Once the blood is withdrawn, they have to shelter them for a while before they can release them back. So it's not typical that they die, but then they need to be monitored for a while before they can be released back into the wild. And it makes sense that they do that because the likelihood for extinction. But that's in the US. There are also some other companies in Europe that do that, but I don't know if they have these regulations in place to see that the horseshoe crab doesn't go extinct. So, yeah.
Thank you. I've seen that picture before, but I have never investigated. No. Other questions for Cynthia? Okay, great. It's nice to see such a complete story, right? So really kind of turning the corner here in terms of people starting to have results, right, and findings. So great. Okay, so I will share again, and we'll talk about this